This scientific session is part of a larger cycle of events that focus on a specific topic, namely the preclinical in vivo tools for the development of large molecule and antibodies. And the reason why JAX is organizing these sessions is because we have recognized that there is a certain need for the scientific community to really develop and discuss these tools in the context of drug development. Large molecules, and specifically antibodies, are the fastest growing group of new drugs in development. And the reason why is that is mostly because of their high specificity, which is likely to lead to less side effects when they're administered to a patient. But these high specificities paradoxically leads to another problem during their development, which is in vivo models are sometimes challenging to use because antibodies recognize human proteins very well, but very often they don't recognize mouse or rat proteins, and sometimes not even monkey proteins. So there really is a need for developing new humanized in vivo models to study these molecules during the preclinical development. Moreover, sometimes the metabolism of these molecules is actively regulated by cellular processes, which might be species-specific as well, like the FcRn that we will be discussing today. The human FcRn interacts, or the human antibodies interact with mouse FcRn in a different ways that with the human. And for this reason, the Jackson Lab really has been working very hard to develop mice that express human or human-like features in different parts of their biology, starting with transgenic FcRn mice to test PK of antibodies or albumin conjugated molecules, to cell humanized mice like PBMC humanized mice or CD34 humanized immune system cell mice to test the immunotoxicity or the efficacy at different stages of the pipeline. Now, today we will be focusing on a specific mouse model, the TG32. You will hear a lot about this model in the next hour or so, which expresses the human FcRn but not the mouse FcRn, and can be used to predict how an antibody, the clearance of an antibody in patients. Delphine will present validation data on the use of this model for bispecific antibodies, and Greg Christensen will focus mostly on the development of different or similar models to cover different applications.